You think you know cell division, mitosis in your high school textbook, 2 cells from 1, clean, simple, done. But there are 7 variations of cell division happening in your body right now that break every rule you learned. Variants that grow hundreds of structures without splitting, cycles that copy DNA but never divide, mechanisms where giant cancer cells butt off daughter cells like yeast, division without spindles, division without nuclei. Your cells are performing spectacles of biological engineering that scientists are still scrambling to understand in 2025. Which of these are keeping you alive, and which ones might kill you? If you value documented cell biology and cellular mechanics history like this, subscribe. It's the best way to support our work. Also comment from which city you're hearing this from. Also, this script is researched and factual, using sources from Nature, published May 2024 by Choksai and Reiter on multiciliated cell differentiation. MIT researchers Goel and Hansen's work in Nature Structural and Molecular Biology from October 2025, showing genome loops persist through mitosis. And the Center for Genomic Regulation's October 2024 publication in Nature Communications on molecular switches controlling division. These aren't undergraduate textbook sources. These are cutting-edge discoveries from the last 12 months showing us cell division is far stranger than we thought. Now back to the information. Number 1, binary fission, the speed demon Escherichia coli. 20 minutes. That's all it takes. Under optimal conditions at 37 degrees Celsius, this bacterial cell copies its entire circular chromosome, elongates its membrane, forms a protein ring, and splits completely into 2 identical daughter cells. 20 minutes from 1 cell to 2. The speed is terrifying when you realize what it means. In 12 hours, a single E. coli can become 68 billion cells if nothing stops it. That's not a typo. 68 billion. Binary fission bypasses every complexity eukaryotic cells deal with. No nucleus to dissolve, no chromatin to condense, no mitotic spindle to assemble. The circular bacterial chromosome sits in a region called the nucleoid cis, attached to the inner membrane near the cell's midpoint. Replication starts at a single origin sequence and moves bidirectionally, both directions simultaneously along the DNA loop. As new DNA is synthesized, each copy attaches to the membrane at different points. The membrane grows, the cell elongates, the chromosomes separate not through elaborate spindle machinery, but through simple membrane growth pushing them apart. The real spectacle is FTZ. This tubulin-like protein discovered in 1991 forms a contractile ring at the division site, the Z ring. FTZ monomers polymerize into filaments that assemble into this ring structure. Anchored by FTZ binding proteins that link it to the membrane, the Z ring recruits more than 30 other proteins to the site, the divisome complex. These proteins coordinate invagination of both the inner and outer membranes, synthesis of new peptidoglycan cell wall material, and remodeling of existing wall. The cell pinches inward from all sides simultaneously. The septum forms 2 cells. But here's what makes binary fission genuinely spectacular beyond its speed. FTZ is homologous to tubulin. They share a common evolutionary ancestor. The protein that bacteria use to divide is the ancient precursor to the protein eukaryotic cells use to build mitotic spindles. The machinery that pulls apart your chromosomes during mitosis evolved from bacterial division machinery. Binary fission isn't primitive, it's the blueprint. Some bacteria push even faster. Certain Vibrio species in nutrient-rich conditions can divide every 10 minutes. 10 minutes. The mathematics of exponential growth become apocalyptic. 1 cell becomes 1 trillion in under 7 hours. This is why bacterial infections can overwhelm immune systems so rapidly, why food poisoning hits suddenly, why sepsis escalates from fever to organ failure in hours. The speed of binary fission is a weapon. The process fails gracefully too. When environmental stress hits, starvation, temperature extremes, DNA damage, many bacteria can pause division while maintaining viability for extended periods. Some enter dormancy, others form spores. The flexibility of binary fission as a survival strategy explains bacterial persistence across nearly every environment on Earth, from deep-sea thermal vents at 400 degrees Celsius to Antarctic ice at minus 89 degrees. One division mechanism, billions of years of refinement, terrifying efficiency. Number 2, mitosis, the precision. Split. Your body performs 2 trillion mitotic divisions every single day. 2 trillion. The stomach lining replaces itself every 3 to 5 days through mitosis. Skin cells divide every 2 to 3 weeks, blood cells continuously. The scale is staggering. roughly 25 million cells completing mitosis every second inside you right now as you hear this. Each division must copy and distribute 46 chromosomes with near-perfect accuracy or risk creating cancerous cells. Mitosis is a 6-phase ballet of molecular machinery. Prophase. Chromosomes condense from loose chromatin into visible X-shaped structures. Each chromosome is already 2 sister chromatids held together by cohesin proteins at the centromere. The nuclear envelope begins to break down. Prometaphase. The envelope fragments completely. Microtubules from the mitotic spindle, assembled from tubulin dimers at the 2 centrosomes, reach into the former nuclear space. Kinetochores, massive protein structures with more than 80 components, assemble at each centromere. Microtubules attach to these kinetochores. Metaphase, chromosomes align along the cell's equator, 46 chromosomes, 92 kinetochores, all precisely positioned in a single plane. The metaphase checkpoint activates. Sensor proteins check every single chromosome to ensure proper bipolar attachment to spindle microtubules from opposite poles. One unattached chromosome can stall the entire process for hours. Anaphase AA, the cohesin holding sister chromatids together, is cleaved by separacy enzyme. 46 chromosomes become 92 individual chromatids. Motor proteins at the kinetochores walk along microtubules toward opposite poles at 1 to 2 micrometers per minute. Simultaneously, anaphase B, the spindle poles themselves push apart as overlapping anti-parallel microtubules slide past each other. Telophase, chromatids reach the poles, decondense back into chromatin, nuclear envelopes reform around each set. Cytokinesis, an actin-myosin contractile ring forms at the cell equator. The ring contracts like a purse string at about 2 micrometers per minute until the plasma membrane pinches closed completely. 2 cells, each with a complete genome. But recent research from October 2025 changed what we thought we knew about mitosis. MIT researchers led by Virat Goel and published in Nature Structural and Molecular Biology discovered that small 3-dimensional DNA loops connecting regulatory elements to genes don't disappear during mitosis, as previously believed. Using higher resolution genome mapping techniques, they found these microcompartments persist through the entire division process. They even strengthen when chromosomes condense. The genome doesn't become a blank slate during mitosis. Structure remains. Regulatory information is preserved and carried through division. Cells have memory built into their chromosome architecture. The checkpoint systems are elaborate, fail-safes. The G1S checkpoint inspects for appropriate cell size and DNA damage before allowing DNA replication. The G2M checkpoint verifies complete DNA replication and checks cell size again before permitting entry into mitosis. The spindle assembly checkpoint, the most stringent, monitors kinetochore microtubule attachments. A single unattached kinetochore generates a weight signal through the MAD and BUB protein families. This signal inhibits the anaphase promoting complex. Anaphase cannot start. Division cannot proceed. Cancer cells often have defective checkpoints. They divide with damaged DNA, with incorrect chromosome numbers. The checkpoints are what keep mitosis safe. When they fail, tumor suppressor genes like p53 can trigger apoptosis, programmed cell death, as a final safeguard. Better to kill a damaged cell than allow cancerous division. Mitotic errors are catastrophic. If chromosomes missegregate, daughter cells receive incorrect numbers, aneuploidy. Most aneuploid cells die. Some survive with profound dysfunction. Trisomy 21, an extra copy of chromosome 21, causes Down syndrome, Turner syndrome, missing an X chromosome, Klinefelter syndrome, and extra X in males. Many miscarriages result from aneuploidy incompatible with life. Mitosis must be nearly perfect across trillions of daily divisions, or the organism fails. Number 3, meiosis, the genetic remix. Meiosis is mitosis's radical cousin. Instead of producing 2 identical diploid cells, meiosis generates 4 unique haploid cells, gametes with half the chromosome number. In humans, meiosis converts a diploid cell with 46 chromosomes into sperm or egg cells with 23. This reduction is essential. When sperm meets egg at fertilization, the chromosome count is restored to 46. Without meiosis, chromosome numbers would double each generation. Within a few generations, cells would collapse under excessive genetic material. Meiosis consists of 2 successive divisions, meiosis 1 and meiosis 2, with only 1 round of DNA replication. Meiosis 1 is the reductional division where homologous chromosomes separate. It begins with a prolonged prophase, one lasting days in females, hours in males. Chromosomes condense in homologous pairs, one from mother, one from father, find each other and synapse together, forming bivalents. The synaptonymal complex, a protein zipper-like structure, holds the homologs together. Crossing over occurs. Sections of DNA between homologous chromosomes break and exchange. Recombination nodules containing enzymes like SPO11 create deliberate double-strand breaks. DNA repair machinery swaps segments. Each crossover creates a chiasma, a physical connection holding homologs together. The average human gamete experiences 50 to 60 crossover events during meiosis, 50 to 60 exchanges between maternal and paternal chromosomes. This generates genetic diversity. Each gamete carries a unique combination of alleles. When you add independent assortment, the random distribution of which homolog goes to which daughter cell, a single human can produce over 8 trillion genetically distinct gametes. 8 trillion. That's why siblings with the same parents are genetically unique, why you are a genetic combination never created before and never to be repeated unless you have an identical twin. Metaphase 1 looks different from mitosis. Bivalents align at the metaphase plate. Kinetochores of sister chromatids fuse into a single functional unit that attaches to microtubules from one pole, monopolar attachment. Homologous chromosomes orient toward opposite poles, bipolar orientation. Anaphase 1, homologous chromosomes separate and move to opposite poles, while sister chromatids remain together. The cell divides. 2 haploid cells, each still with duplicated chromosomes, 2 sister chromatids per chromosome. No S phase occurs between meiosis 1 and 2. Meiosis 2 resembles mitosis, but starts with haploid cells. Prophase 2, chromosomes condense again. Metaphase 2, chromosomes align. Anaphase 2, sister chromatids finally separate. Telophase 2 and cytokinesis, 4 haploid cells result. In males, all 4 become functional sperm. In females, cytokinesis is asymmetric. One cell receives most of the cytoplasm, becoming the egg. The other 3 become polar bodies that degenerate. This asymmetry ensures the egg has sufficient cytoplasmic resources and organelles to support early embryonic development. Meiotic errors are common and devastating. Non-disjunction, failure of chromosomes to separate properly, causes aneuploidy in gametes. If a gamete with an extra chromosome 21 fertilizes a normal gamete, the embryo has trisomy 21. Maternal age dramatically increases meiotic error rates. At age 20, a woman has about 1 in 1,500 chance of conceiving a child with Down syndrome. At age 40, 1 in 100. At 45, 1 in 30. The mechanisms maintaining accurate meiotic chromosome segregation degrade with age. Oocytes arrest in prophase 1 before birth and remain there for decades, 10 years, 20 years, 40 years, before completing meiosis at ovulation. The cellular machinery maintaining chromosome integrity slowly fails. Number 4, amitosis, the chaotic splitter. This is where cell division gets genuinely strange. Amitosis, also called direct cell division, involves nuclear budding without any of the elaborate chromosome choreography of mitosis or meiosis. No spindle forms, no chromosomes condense and align. The nucleus simply elongates, constricts in the middle league and splits. The cytoplasm divides. 2 cells. The DNA distribution is essentially random. Daughter cells receive unpredictable amounts of genetic material. It sounds like cellular chaos. It is. And it's happening inside you. Amitosis was first described in the 1870s in ciliate protozoans. Scientists dismissed it as pathological, defective division in stressed or dying cells. They were wrong. Amitosis is a legitimate developmental program in specific cell types. It occurs in placental trophoblast giant cells in mammals, in polyploid cells of plant endosperm, in certain ciliated protozoans where macronuclei divide amitotically while micronuclei divide mitotically. The mechanism is reproducible and regulated, not random cellular failure. The most spectacular and terrifying amitosis occurs in cancer. In the early 2000s, researchers Kirsten Wallen and Rangaswamy Rajaraman independently reported that polyploid giant cancer cells could produce rapidly proliferating daughter cells via amitotic division, a process termed meiosis. When chemotherapy or radiation damages cancer cells, some survive and become polyploid giants, PGCCs, with multiple copies of their genome in a single enormous nucleus. These cells can reach 32C, 64CI, even 128C DNA content where C represents the haploid genome. Normal diploid cells are 2C. Then something remarkable happens. The giant polyploid nucleus undergoes asymmetric division through nuclear budding. Small buds containing portions of nuclear material pinch off. These budded nuclei separate from the parent. Cytokinesis follows. The small budded cells, called karyoplasts or Raju cells after researcher Raju who described them, contain variable amounts of DNA, often near diploid or paradiploid levels. These karyoplasts can re-enter the mitotic cycle and proliferate rapidly. A single PGCC can produce hundreds to thousands of daughter cells through repeated amitotic budding. The daughter cells often exhibit stem cell-like properties. They can be highly metastatic. They resist therapy. They repopulate tumors after treatment. Studies published in September 2024 in the journal Cancers documented this mechanism in detail. PGCCs arise after treatment with DNA-damaging agents. They remain viable and metabolically active. They undergo depolyploidization through amitosis and a meiosis-like process. Yes, some cancer cells activate meiotic genes. The daughter cells emerging from PGCCs contribute to tumor recurrence and therapy resistance. The initial tumor and the recurrent tumor post-treatment are different diseases requiring different strategies. Amitosis in cancer isn't pathology, it's adaptation, it's survival, and we're still figuring out how to stop it. The mechanism of amitotic division remains poorly understood. No cohesin cleavage, no separase activation, no anaphase-promoting complex. How does the nucleus split? How is DNA partitioned? Research suggests the condensed pterionae, duplicated chromosomes and polyploid nuclei may maintain some organizational structure that facilitates division. But the details are murky. What's clear is that amitosis exists, it's functional, and in the context of cancer, it's a significant threat to treatment success. If you value documented cellular biology and cancer research like this, subscribe. It's the best way to support our work. Also, comment from which city you're hearing this from. Also, this deep research includes sources from the journal Cancers. September 2024, issue by Mirzayans and colleagues reviewing amitotic cell division and therapy resistance. Not general cancer textbooks, but specialized research showing exactly how these polyploid giant cells evade treatment through bizarre division mechanisms. Now back to the information. Number 5, end-or duplication, the DNA duplicator, without division. Some cells copy their genome repeatedly without ever dividing. They just keep replicating DNA. 2 copies become 4, 4 become 8. 8 become 16, 32, 64, 128, all the way up to incredible ploidy levels. The highest recorded is 24,000 C in some plant endosperm cells. 24,000 copies of the genome in a single cell. That's not mitosis gone wrong. It's a deliberate developmental program called end-or duplication or endocycling. The endocycle consists only of alternating G and S phases, no mitosis. Cells grow, replicate their DNA during S phase, then enter a gap phase preparing for the next round of DNA synthesis. They bypass mitosis entirely. The result is polyploid cells, cells with multiple complete sets of chromosomes that grow dramatically larger than diploid cells. The cell size generally scales with DNA content. A cell with 16 copies of the genome is substantially bigger than a cell with 2 copies. End-or duplication is widespread. In Drosophila, nearly all differentiated tissues use it. Salivary gland cells reach 1024 C, 1024 copies of the genome. The chromosomes remain as polytene chromosomes where all sister chromatids stay aligned in parallel, creating the famous banded appearance under microscopes that enabled early genetic mapping. In plants, end-or duplication is even more common. It occurs in leaf cells, root cells, fruit cells. The majority of plant tissues show some degree of polyploidy. Tomatoes, wheat, strawberries, polyploid cells. In mammals, end-or duplication is more specialized but equally important. Megakaryocytes, the bone marrow cells that produce blood platelets, undergo endomitosis to reach up to 128 C. During endomitosis, cells enter mitosis, chromosomes condense, the nuclear envelope breaks down. But then, mitosis aborts before nuclear division. Chromatids separate but aren't distributed to daughter cells. They remain in a single nucleus when the envelope reforms. The cell re-enters G1 and another round of DNA synthesis. The megakaryocyte gets bigger with each endomitotic cycle. The large polyploid cell fragments into thousands of platelets, the cell pieces that form blood clots. The polyploidy achieved through endomitosis is essential for sufficient platelet production. Without it, humans would bleed out from minor injuries. Mammalian trophoblast giant cells in the placenta use end-or duplication to reach 512 C in mice. These giant polyploid cells provide the barrier between maternal blood and fetal tissue. Their size is crucial to their function. Recent research showed rodent trophoblast giant cells undergo true endocycling, no mitotic features, while megakaryocytes undergo endomitosis with abortive mitosis. Both produce polyploidy, but through slightly different mechanisms. The distinction matters less than the outcome. Big cells with multiple genome copies serving specialized functions, Why endoreplicate instead of just making more cells through mitosis? Several reasons. First, speed. Endocycling is faster than mitosis. No time wasted assembling spindles, condensing chromosomes, performing cytokinesis. Just replicate DNA and grow. Second, metabolic capacity. Polyploid cells have more gene copies, so they can produce more mRNA and more protein. High metabolic output. Third, terminal differentiation. Endoreduplication is often irreversible. Polyploid cells rarely divide again. This permanence is useful for cells that need to stay put and perform one function indefinitely. Fourth, stress response. Plants use endoreduplication as a survival mechanism. DNA damage in Arabidopsis root cells triggers downregulation of mitotic factors and promotes endoreduplication. The cells copy their genome to maintain genetic redundancy rather than risk dividing with damaged chromosomes. But endoreduplication has constraints. Mitotic division of endopolyploid cells is rare. Once a cell is polyploid, it's locked in, the plasticity is lost, and there's significant metabolic cost. DNA replication requires phosphorus and nitrogen, nutrients often limited in natural soils. Studies show polyploid plant species correlate with high soil phosphorus availability. The genome has to be worth replicating multiple times. The molecular control of endoreduplication involves cell cycle regulators repurposed. Cyclin E and CDK2 activity is maintained to drive S phase. Cyclin B and CDK1 activity, the mitotic kinases, is suppressed. The anaphase promoting complex activated by CDH1 degrades mitotic cyclins. String phosphatase, also called CDC35, which normally activates mitotic cyclin CDK complexes, is repressed. The cell has enough S-phase activity to replicate DNA, but not enough M-phase activity to enter mitosis. It's stuck in a loop of DNA synthesis and growth. Deliberate, controlled, spectacular. Number 6, the multiciliation cycle, the centriole factory. This is the newest entry on the list and possibly the most spectacular. In May 2024, researchers Semil Chokshi and Jeremy Ryder at the University of California, San Francisco published a groundbreaking discovery in Nature, issue 630, pages 214 to 221. They found that cells in mammalian airways, brain ventricles, and reproductive tracts use an entirely new variant of the cell cycle to become multiciliated. The multiciliation cycle. These cells need to grow hundreds of motile cilia, hair-like projections that beat in coordinated waves to move mucus in lungs or cerebrospinal fluid in the brain. Each cilium requires a basal body to nucleate it. Each basal body is a matured centriole. Normal cells have 2 centrioles. Multiciliated cells need hundreds. So what do? They hijack the cell cycle machinery. When Chokshi and Ryder used single-cell RNA sequencing on differentiating multiciliated cells from mouse lungs, they found expression patterns that looked like the mitotic cell cycle, sequential expression of cyclins and cyclin-dependent kinases matching G1, SA, and G2 M phases. But these cells weren't dividing. DNA replication didn't occur. The cells didn't split. Instead, they produced hundreds of centrioles. The cell cycle regulators, proteins that normally coordinate DNA replication, chromosome segregation, and cytokinesis, were being redeployed to control centriole amplification and cilia formation. The multiciliation cycle proceeds through 4 stages. Stage 1, precursor cells express early transcription factors like MYB and upregulate proteins for centriole biogenesis. Stage 2, specialized structures called deuterosomes appear. These are non-membranous organelles unique to multiciliated cells. Deuterosomes generate new centrioles, lots of them, 30 to 500 centrioles depending on cell type. Stage 3, centrioles disengage from deuterosomes, migrate to the cell surface, mature into basal bodies by acquiring distal appendages marked by protein CEP164, and dock at the apical membrane. Stage 4, ciliogenesis. Each basal body extends a cilium. The cell is now covered with hundreds of beating cilia. The key difference from the normal cell cycle is E2F7. This transcription factor is expressed at low levels in proliferating stem cells but surges in multiciliated cells. E2F7 suppresses DNA synthesis during the S-like phase of the multiciliation cycle. It promotes the transition to the G2M-like phase. During this phase, cyclin B1CDK1, the master mitotic kinase, promotes growth of newly forming centrioles instead of driving mitosis. The anaphase promoting complex controls centriole number. CDK2 promotes the final differentiation stage. It's the same molecular orchestra playing a completely different tune. When the researchers knocked out E2F7 in mice, multiciliated cells failed to differentiate correctly. The knockout cells showed increased DNA synthesis markers. They tried to replicate their genomes like normal dividing cells. Hundreds of centrioles got stuck in the cell body instead of migrating to the surface. The mice developed hydrocephalus, fluid buildup in the brain, because multiciliated ependymal cells lining brain ventricles couldn't clear cerebrospinal fluid without functional cilia. Without E2F7, the multiciliation cycle collapses into something closer to the canonical cell cycle, and the cells fail their specialized function. This discovery revealed something profound. The cell cycle isn't just for making two cells from one. It's a modular toolkit. Evolution can repurpose its components for different cellular projects. The same regulatory proteins that prevent cancer by limiting centriole number and dividing cells are deliberately overridden in multiciliated cells to produce hundreds of centrioles. It's a controlled break of cancer prevention rules to achieve a developmental goal, and we only learned about it in 2024. There's likely more cell cycle variants we haven't discovered yet. Writers suggested as much. Maybe lots of cells use variant cell cycles to drive differentiation. A multiverse of cell cycles. We're just starting to map it. Number 7. Budding. The asymmetric split. Saccharomyces cerevisiae, baker's yeast, divides by budding. Instead of splitting symmetrically like mitosis, the mother cell grows a small protrusion called a bud. The bud enlarges. A nucleus migrates into it. Eventually, the bud separates, becoming a daughter cell. Asymmetric division. The mother cell retains its identity and can bud repeatedly. The daughter cell starts small and must grow before it can bud. This asymmetry creates age structure in yeast populations. Mother cells are replicatively aged. They have a finite budding capacity, typically 20 to 30 buds before senescence. Daughter cells are young. Budding is mitotic division with spatial asymmetry. The yeast cell cycle is similar to other eukaryotes, G1, Si, G2, M phases. Bud emergence occurs in late G1, triggered by cyclin C-LN activity. The site of budding is determined by cortical markers, proteins that localize to a specific spot on the cell surface. In haploid cells, budding occurs adjacent to the previous bud site, axial budding. In diploid cells, budding alternates between the two poles, bipolar budding. Bud site selection proteins including BUD1, BUD2, BUD5, and the landmark protein BUD3 ensure proper positioning. The bud grows throughout S and G2 phases. Actin cables and myosin motors transport vesicles and organelles from the mother into the growing bud. The nucleus remains in the mother cell initially. During mitosis, the nucleus elongates and spans the neck between mother and bud. Spindle microtubules position one set of chromosomes in the mother, one in the bud. Nuclear envelope does not break down. Yeast undergo closed mitosis, where the nucleus remains intact throughout division. Anaphase, chromosomes segregate. One nucleus remains in the mother. One moves completely into the bud. Cytokinesis occurs at the narrow neck connecting mother and bud. A septum forms, similar to bacterial division, and the cells separate. The asymmetry is functional beyond size. Mother cells accumulate damage over successive budding cycles. Oxidatively damaged proteins, extrachromosomal ribosomal DNA circles, dysfunctional mitochondria, these are retained in the mother cell and excluded from buds. Quality control mechanisms ensure daughters receive functional organelles and minimal damage. This is why daughters are rejuvenated. They start fresh, mother's age. This aging isn't random. It's a strategy to protect daughter cells from inherited damage. But budding isn't exclusive to yeast. Hydra, the small freshwater polyp, reproduces asexually through budding. Cells on the body column undergo mitosis and form a mass. The bud grows into a miniature hydra complete with tentacles. Eventually it detaches and becomes an independent organism, coral polyps bud. Some plants propagate through budding. Potato eyes are buds. And terrifyingly, cancer cells can bud. Polyploid giant cancer cells undergoing amitosis produce small buds that pinch off. Budding in cancer combines the asymmetry of yeast budding with the genomic chaos of amitosis. Small, rapidly proliferating. Therapy-resistant daughter cells emerge from giant, damaged parent cells. The molecular machinery of budding involves formins, proteins that nucleate actin filaments, and septins, GTP-binding proteins that form filaments at the mother bud neck. Septin rings scaffold the division site and serve as diffusion barriers, ensuring mother and bud maintain distinct protein compositions. The myosin motor myo1 forms a contractile ring that constricts the neck during cytokinesis. Chitin synthase deposits cell wall material to seal the septum. The division is asymmetric in size and content, but symmetric in genetic material. Each cell receives a complete genome. Budding demonstrates that cell division doesn't require equality. Asymmetric division generates cellular diversity. In stem cell populations, asymmetric division produces one daughter that remains a stem cell and one that differentiates. Neuroblasts in Drosophila divide asymmetrically to generate neurons. Skin stem cells divide asymmetrically to maintain the stem cell pool while producing differentiated keratinocytes. Asymmetry is a feature, not a bug. Budding is the clearest example. One cell gives rise to two. But they are deliberately different from the start. The 7 types of cell division, binary fission, mitosis, meiosis, amitosis, end-or duplication, the multiciliation cycle, and budding represent fundamentally different strategies for the same basic goal: perpetuate acellular or organismal life. Some prioritize speed, some prioritize genetic diversity, some prioritize size. Some adapt existing machinery for new purposes. Some break rules that normally prevent cancer. All are happening right now across the tree of life in bacteria, protists, fungi, plants, and animals, including inside you. The story doesn't end with 7. Cell division is more diverse than this list captures. Multiple fission, one cell splits into many simultaneously, occurs in some parasitic protozoans like Plasmodium causing malaria. Schizogony in these parasites produces dozens of daughter cells from a single mother in one division event. Plasmotomy, multinucleate cells undergoing cytokinesis without nuclear division, occurs in certain ciliates. Fragmentation in multicellular organisms like planarian worms allows body pieces to regenerate into whole organisms through specialized division programs. Budding variations exist across sponges, echinidarians, and even some multicellular plants. But here's what makes these 7 spectacular. They're not exotic curiosities found in obscure organisms. These are core mechanisms. Binary fission in the bacteria that live in your gut, mitosis in your skin cells right now, meiosis that produced the gametes that created you, amitosis in your placenta during development and possibly in cancer cells if you're unlucky, and/or duplication in your liver cells and megakaryocytes. The multiciliation cycle in your airway epithelium clearing the mucus you produce, budding in the yeast that made your bread or beer. These divisions are personal. They're happening to you or because of you, and we're still learning. The multiciliation cycle was discovered in May 2024. What else don't we know? How many other variant cell cycles exist? Do neurons use modified cell cycles to extend processes? Do muscle cells use variants to become multinucleate syncytia? How do giant neurons in squids and whales coordinate nuclear divisions? How do slime molds coordinate thousands of nuclei dividing in a common cytoplasm? The deeper we look, the stranger it gets. What researchers found in October 2025 changes our understanding of mitosis itself. Those persistent microcompartments discovered by MIT researchers, the fact that regulatory DNA loops survive division and even strengthen during chromosome condensation, means cells have structural memory through division. Information encoded in 3-dimensional genome architecture passes from mother to daughter. Mitosis isn't a reset. It's a carefully controlled inheritance of not just DNA sequence, but DNA organization. The implications for development, for cell identity, and for cancer are profound and still being worked out. And what about the control systems? How do cells decide which division type to use? Diploid cells undergoing stress can enter end-or duplication instead of mitosis. Cancer cells after chemotherapy can switch from mitosis to amitosis. Stem cells can shift from symmetric to asymmetric division. The choice between division types isn't hardwired. It's regulated by signaling pathways, transcription factors, and cell cycle checkpoints. We're starting to understand the decision trees: cyclin levels, checkpoint protein activity, transcription factor, expression. These are the switches that route cells down different division paths. The Center for Genomic Regulation published work in October 2024 in Nature Communications, showing researchers created a molecular switch using the protein PRC1 that can control cell division on demand outside a living system. By manipulating phosphorylation of PRC1, they induced large-scale transitions between different states of cytoskeletal organization needed for division. The changes took only minutes. This is the beginning of division control, the ability to turn division on and off, to push cells toward one division type or another. The therapeutic potential is obvious. Cancer is uncontrolled division. What if we could force cancer cells into end-or duplication, where they grow huge and polyploid but can't proliferate, or push them into terminal differentiation, or trigger catastrophic mitotic failure? Division control is the next frontier. But it's not just about stopping bad division. It's about enabling good division. Regenerative medicine needs controlled division. Growing organs from stem cells requires precise control over when cells divide, how they divide, whether they differentiate or stay pluripotent. Tissue engineering needs cells that divide and organize correctly. Understanding division variants like the multiciliation cycle tells us how to direct cells to produce specific structures. How to make airways that actually work. How to grow functional kidney tubules with proper epithelial architecture. How to engineer tissues that aren't just cell masses but organized functional organs. The molecular details matter. The fact that E2F7 switches cells from the canonical cell cycle to the multiciliation cycle means we can manipulate E2F7 to control cilia formation. Patients with ciliopathies, genetic diseases affecting cilia, have defective multiciliated cells leading to chronic respiratory infections, infertility, hydrocephalus, kidney disease. If we understand the multiciliation cycle well enough, we might be able to rescue defective cilia production. Gene therapy to restore E2F7 function, small molecules that enhance centriole amplification. Cell-based therapies using correctly differentiated multiciliated cells grown in culture and transplanted to repair damaged airways. The biology informs the medicine. The same is true for cancer. Understanding amitosis and how polyploid giant cancer cells produce therapy-resistant daughters means we can target that process, inhibit nuclear budding, block depolyploidization, prevent karyoplast formation, force PGCCs into senescence or apoptosis. The current problem is that conventional chemotherapy and radiation create PGCCs. They're a consequence of treatment. We kill most cancer cells, but survivors become polyploid giants that later repopulate the tumor through amitosis. If we understand the mechanism, we can block that escape route. Combination therapies that hit dividing cells with conventional agents and hit PGCCs with amitosis inhibitors. Different diseases require different strategies. The researchers were right about that. 7 types of cell division. 7 strategies for perpetuating life. 7 mechanisms refined over billions of years of evolution. 7 processes you're now aware of happening all around you and inside you. Your gut bacteria dividing every 20 minutes through binary fission. Your blood cells dividing every few days through mitosis. Your gametes preparing for potential fertilization through meiosis. Your megakaryocytes growing huge through indoor duplication to make the platelets that will clot your next cut. Your airway cells using the multiciliation cycle to produce the cilia that clear your lungs every time you cough. The asymmetric divisions in your skin-maintaining stem cell populations. And in the background, the shadow, amitosis in cancer cells if they emerge, budding off resistant daughters, evading treatment, threatening survival. Here's what ties them together. Every division type is a solution to a specific problem. Binary fission solves for speed and simplicity. Mitosis solves for genetic accuracy and controlled growth. Meiosis solves for genetic diversity and sexual reproduction. Amitosis solves for survival under extreme stress in polyploid cells. Endoreduplication solves for cell size and metabolic capacity without the cost of division. The multiciliation cycle solves for producing hundreds of specialized structures without losing cell identity. Budding solves for maintaining youth in daughter cells while sequestering damage in mothers. Different problems, different solutions. Same underlying goal, continue existence. But evolution doesn't engineer perfection, it satisfices. It finds solutions that work well enough given the constraints. Binary fission is fast but vulnerable to antibiotics. Mitosis is accurate but slow. Meiosis generates diversity but causes aneuploidy when it fails. Amitosis enables cancer survival. Endoreduplication is irreversible. The multiciliation cycle breaks cancer prevention rules in a controlled way that sometimes goes wrong. Budding creates aging mothers. Every solution has trade-offs. Every mechanism has failure modes. The fact that life works at all given these imperfect processes is remarkable. And the fact that we've discovered a new division variant in 2024, the multiciliation cycle, after studying cell division intensively for over 150 years, since Walter Fleming coined the term mitosis in 1882, tells you how much we still don't know. Writer said it, evolution has adapted the cell cycle to carry out a variety of cellular projects well beyond cell division. We've mapped 7. How many more are there? What other variant cycles are operating in specialized cells we haven't looked at closely enough? What happens in giant neurons? What happens in syncytial muscle? What happens in plant cells with bizarre ploidy patterns? What happens in organisms we haven't studied yet? Cell division is not a solved problem. It's an active research frontier. The discoveries keep coming. MIT's finding about persistent genome loops in October 2025. The Center for Genomic Regulation's molecular division switch in October 2024. Umia University's work on mediator protein movement controlling division timing published May 2024 in Science Daily. The discoveries about PRC1 protein preventing chromosome breakage during division from HKU researchers in January 2025. Every month brings new findings. Every paper reveals another layer of complexity. Every technique, single-cell sequencing, super-resolution microscopy, genome editing, in vitro reconstitution, shows us something we couldn't see before. The stakes are high. Cancer is out-of-control division. Infertility is failed meiosis. Chronic infections stem from defective multiciliated cells with faulty division programs. Tissue damage that won't heal is inadequate mitotic response. Aging might be accumulated division errors. Every major category of human disease touches cell division somehow. Understanding division isn't academic, it's medical, it's personal. So, which of the 7 types of cell division will you remember? The speed demon binary fission making 68 billion bacteria from 1 in 12 hours. The precision split of mitosis performing 2 trillion perfect divisions daily in your body. The genetic remix of meiosis that made your unique gametes. The chaotic amitosis and polyploid giant cancer cells budding off therapy-resistant daughters. The DNA duplicator Endor duplication making megakaryocytes 16 times bigger to produce your platelets. The newly discovered multiciliation cycle building hundreds of centrioles in your airway cells. The asymmetric budding segregating damage away from daughter cells. All 7 are spectacular. All 7 matter. All 7 are happening right now. A recent excavation at the site of the old cell biology building at Rockefeller University found Walter Fleming's original notes from 1880 describing what he called karyokinesis, nuclear division. In those notes, he sketched dividing cells from salamander embryos. The drawings show condensed chromosomes, spindle fibers, daughter cells. He saw it all 145 years ago with primitive microscopes and no understanding of DNA. The drawings are accurate. The process he saw is the same mitosis happening in your cells right now. 145 years of research, and we're still finding new variants, still discovering mechanisms, still learning how cells split. Division is life. Every organism that ever lived descended from cells that divided successfully. Every cell in your body traces back through an unbroken line of successful divisions to the origin of life itself 3.8 billion years ago. 3.8 billion years of division without a single failure in your direct lineage. That's the real spectacle. Not just that cells can divide in 7 different ways, but that division works reliably enough, accurately enough, flexibly enough to build and maintain every living thing on Earth, from bacteria to blue whales. That through all the variants, all the mechanisms, all the spectacular differences, the fundamental act of one cell becoming 2 or 4 or hundreds has been preserved and refined for billions of years, and that we're alive right now, in 2025, still discovering new ways that cells accomplish this ancient, essential, spectacular process. Every division is a beginning. Every split is a continuation. Every cell that completes division successfully carries forward the unbroken chain. The 7 types aren't just mechanisms, they're the toolkit that made you possible.